Hi everyone, this is a brief video on the reverse transcriptase or the reverse transcription procedure of HBV, how it works. Okay, first we will see what is the genome structures, like basically from pgRNA. Okay, from pgRNA, which looks like this. Okay, this is basically a pgRNA that is transcribed from the cccDNA. Remember, I have already mentioned in my previous short videos. If you missed it out, so you can go and check my channel. I have already added it in my shorts. So basically, in this case, what happens? This is the stem loop structure, which is known as epsilon. Okay, this particular stem loop structure is known as epsilon at the 5' end of the pgRNA. Now here there is a sequence that is known as DR1. Now DR stands for direct repeat. Okay, DR1. Then here there is a structure which is 3' proximal end of the pgRNA, that is DR1 star. That is the copy of this DR1 but has few structural changes which differentiate this specific sequence from this one. Okay. Another thing, that is about 30 nucleotide upstream from this DR1 star sequence, there is a sequence called psi. Okay, there is a sequence called psi, and there is another sequence that is just overlapping of this DR1 star sequence, that is called omega. Now all these 5 sequences, particular specific these sequences, specifically this epsilon, psi, DR1 star and omega, all these 4 sequences specifically, they have functions, important or essential functions to initiate the reverse transcription process from pgRNA that has been already transcribed from cccDNA in the nucleus. Now this pgRNA, that is basically acts as a template to produce the oligo-DT. Okay, now this oligo-DT uses this bulged region of epsilon structure as a template to make a short primer. Okay, so this is basically a primer, and this bulged region of the epsilon is the template. Remember that this is a template. This is for all PG same for virology syllabus. It's a very important topic. Guys, please, you should learn about it, because these type of questions, that the initiation process or the jumps, what are the jumps or what are the procedures of the jumps or template switching occurs in case of reverse transcription process of particularly HBV. These type of questions are very common and very important in virology question papers. You can check into the university website of the Calcutta University as well, and this is for the PG same 4 syllabus. It's a very important topic, so I should continue this thing. So in this case, this is basically the primer and this one is the template. Okay, so the template is basically pgRNA and this one is the primer. Now this is made by the reverse transcriptase enzyme, which is the P protein that is translated from this pgRNA. Okay, remember I have already mentioned in this or in my previous video that after being synthesized or translated and after its post-translational modification, P protein okay, they will bind to its own template at the epsilon region. So here it will bind like this. This is basically the RT region. RT domain means the reverse transcriptase domain. There is a small RH means the RNase H domain, and there is the main domain which actually make base pair with this primer, newly made primer, that is the TP domain, means the transfer protein domain. Okay, now there is a linker region also in between these two. Now one thing you have to remember, that there is also a TP domain here. Okay, this part, this is the integral part of this whole P protein. This is a P protein. Okay, this is a P protein. So now what happens, to initiate the minor strand of DNA from this pgRNA, that is why this process is known as reverse transcription, because here the RNA is the template from where the DNA has been made. That is why it is just the reverse of the transcription, because transcription means RNA will be made from RNA, but here the DNA will be made from RNA. So to start or initiate the small size of minor strand DNA of the RC DNA, remember that is the main genome of HBV in the virions, they need a primer. So this is the first step, the primer has been made. Now one thing that is very important, this primer region that is linked to the TP domain of this P protein, because this TP domain has tyrosine 63, means at the 63 position in the TP domain there is a tyrosine residue. Now this tyrosine residue that will make a bond with this primer, and that will drag this whole primer from here to here, this DR1 star sequence. Now this primer that will bind this DR1 sequence like this. Okay, so this is the first primer template switch or primer jump. There are total 3 jumping stages in the HBV reverse transcription, but here this is the first or the initial jump of the primer from this epsilon bulged region to DR1 star. Remember DR1 star. If you just right to forget this star, it means it's DR1. So this will be completely wrong. So DR1 star. Okay. Now DR1 star has specific acceptor site. That is why this primer that will bind to this specifically. But here this psi and this omega make this binding much more specific. Okay. So with this, the specificity of this primer switch to this a new template, that will elongate the minor strand of the DNA. Okay. So in this way, the initiation of the reverse transcription occurs, and in my next videos, I'll definitely continue this topic, how the second and third jump occurs and how the complete minor strand DNA has been synthesized, and incomplete plus strand that will also be synthesized, and both of them will make a RC DNA. Okay, so this is all. If you like this video, please subscribe my channel and share with your friends. Thank you so much. Have a good day.